Okay. So today's speaker is Dr. Matt Allender. I'm going to let him introduce himself to you all today. So welcome, Matt, and I will turn it over to you.
All right, thanks, Aaron. Okay, thanks everybody. It's really great to be here. Hopefully, over the next 45 minutes or so, I'll give you a little insight into how we're really saving the world one box turtle at a time. It'll be a little sciencey today, but I'm going to frame it as a story. So hopefully everybody we'll enjoy that aspect. I don't want to get too sciencey. I know there's a diverse crowd, and my mom, I'm sure, is on this call. She was a elementary school teacher for a while, so my goal is to make sure that everyone with every background is able to kind of follow along. But a little bit about myself. So I'm a zoo and wildlife veterinarian. I have been at the university for over 16 years now. And well, as a faculty member, I actually have 5 degrees from the University of Illinois. That wasn't a typo. It's kind of crazy. But zoo and wildlife medicine is really broad, right? Like when we go to the doctor, we'll go to a dermatologist or a cardiologist, various specialists. But zoo and wildlife medicine is all of these things. In fact, most of the large animals that I see, a tiger or a lion, half the job that I do is keeping it asleep safely. And so I do a lot of anesthesiology. Um, and then we have to be pretty knowledgeable about all of these different um, disciplines for roughly 75,000 species. And that doesn't even include inverts. And we've done some blood studies, hemolymph studies in several species of invertebrates. So it is really broad. It's kind of odd sometimes when I'm training our residents that we're specializing in really being a generalist because we've got to know just about everything except for in humans, dogs, cats, cows, horses. And we have to know all of that stuff in order to be good at many of the things that we're doing. So how did I get to where I'm at? Just again, a little background before we get into turtle stuff is I grew up in Champaign-Urbana, so I am a townie, certainly. And then I did my first degree, my first bachelor's in ecology, ethology, and evolution at Illinois. I got my vet school from Illinois, as well as a master's in conservation medicine, looking at mainly rattlesnakes, Massasauga rattlesnakes. I was in private practice, so I was like your regular dog and cat vet in Urbana for a couple of years before I went in and did a residency in zoological medicine at the University of Tennessee and the Knoxville Zoo. And I was there for 3 years specializing. I came back after that, joined the faculty, and concurrently did a PhD in epidemiology, looking at emerging diseases of turtles, and one of them we're going to talk about today. And then I joined the faculty full time in 2009. And then about a little over 5 years ago, I became the director of conservation medicine and science at Brookfield Zoo Chicago in a joint program where I'm also director and founder of the Wildlife Epidemiology Lab at Illinois. So I wear a lot of different hats. I get to do a lot of amazing things. I really love my job. And largely because, especially as we're coming out of the summer field season, I get to spend most of my summer working with turtles and students that are working with turtles. So it's pretty wonderful. But let's make that as a transition and let's start the story of of again what the 16 years of college and training. So that was after high school to get to this point. Where's it really led? All right, so we're going to start this story with North American box turtles. Box turtles, and especially Eastern box turtles, are my favorite species. I enjoy working with them. They're quite charismatic individuals. They're stubborn. They've got individual personality, certainly, and they're really a great species as a sentinel for environmental and ecosystem health. So box turtles are both live in terrestrial and aquatic environments, so they rely on those habitats just like people do. They're omnivorous, just like people are. They're long-lived. Again, some box turtles can live over 150 years. Um, and so they're quite long-lived, and they only live in small areas. So usually their entire range for their life is takes up at about 5 to 15 acres. So yeah, that's about it. And so their health is a direct reflection of the health of the ecosystem that those animals are found in. So if they're unhealthy, then that ecosystem is unhealthy, and then we have opportunities in order to fix it. Now, as a veterinarian, I spend my time looking at health. I spend a lot of time looking at diseases, but that can look in a lot of different ways. So box turtles can die, and we've done several studies, and this again, I'm just going to tell a story. I'm not really going to explain a lot of the graphics, but each one of those circles accounts for the size of the population of turtles that we studied over a period of time, from April through September, from 2011 to 2015. And the color references whether those animals were experiencing outbreak of disease, the dark color, or a before or after the outbreak. And so animals can die in the wild, and it doesn't have to just be natural. Many of these animals were dying due to diseases that were associated with bacterial or viral infection. But when we find dead turtles in the wild, oftentimes they've decomposed to the point where there's only bone. And so we've developed new techniques, things like looking at the bone marrow of just shells that have been recovered in the woods to try to look to see if there's viruses that have been circulating in the blood to give us an idea of what's going on and what's causing those outbreaks. And things like freezing events certainly have led to death. And so as populations are experiencing really climate change driven weather events, things where, we've got an event in February where it's 75 or 80 degrees and they're out for a little bit. And then in March, it hits, freezing temperatures again. Those animals have come out and then they don't have enough time in order to get back down. And so those freezing events can be pretty impactful depending on the year. Things like reproductive stasis, where their eggs don't develop, and that's from numerous different habitat or infectious causes. So historically, and this has changed this year, I've used a team of both volunteer vet students, which is not shown here, and a team of canines. And as you'll see here, these dogs will work in this coordinated pattern where they're searching through the environment to find really cryptic species. And in order to be able to find turtles in this environment, like you can't see that, right? And people it would be really hard for, but dogs can do it. Dogs can sniff it out and bring them right to us. We hike with them, or we did, and they're really going through that dense prairie in order to be able to find animals for us to be able to um, do physical exams and their wellness checks. So they're a pretty big tool. And so over the course of the last 17 years, we have done health assessments on over 5,000 box turtles. It's the largest health study of box turtles anywhere in the world. And so when we do that, we've taken that opportunity in order to look at what is the impact and what is the rate of using dogs and humans in order to find turtles, and how does that affect those animals both physiologically and through muscular contractions. We've developed lots of different techniques in order to validate. So if you think about it, right, when you go to the doctor, when you take your dog to the doctor, those are really common species, humans and dogs. And there's been a lot of data over the course of the last hundreds of years that tells us what normal is. Well, we don't really know what normal is for box turtles, and they're ectothermic, so they're impacted by the environmental temperature. So their health in April is very much different than their health in July because the temperatures are a lot different. And so we've done a lot of that validation. We do normal morphometrics and demographics. So like, again, when we go to the doctor, we stand on the scale and we get measured. Well, that's what these guys do as well, because certain diseases or health factors are associated with their size or with their age class or with their weight. And we want to know that they have good body condition. So just like when we stand on a scale, there could be a human that weighs 180 pounds that is perfect weight. somebody that weighs 180 pounds that is obese, somebody that weighs 180 pounds that is underweight. And so weight by itself is not really a good indicator of health and wellness. And similarly in humans, right? Like you measure the body mass index. It's an equation based on weight and height that tells you what your body mass index is. Well, that didn't exist for turtles, but we sent 65 box turtles through the CT scanner and we characterized their fat content, and then we related it to different weight and measurements. And then we could develop this scale. So many of you parents out there will recognize something like this, right? You can go over to the age, and in humans this is months usually, but age for turtles, and then you can go up to their weight and you can figure out, are they in the 25 percentile? Are they in the 75 percentile? And this helps us with obviously conservation, because if certain habitats are supporting turtles of much lower body condition, well, then that's something that we really want to know. Something's going on in that environment that we might need to fix. And then we do physical exams. So things like this white tip of the tongue in this box turtle is completely abnormal. It's a plaque, is what we call that, and it's caused by a viral infection. Or if you look at the nose, eyes, ears, nose, throat, the nose, this nair is smaller than this nair. And we see that typically after they've recovered from bacterial infections. We look at their shell. There's lots of different things that cause abnormalities with their shell. We can look for a lot of different physical exam abnormalities, such as infections in the ears or clear discharge from the nose or eyes. All of these things are part of our physical examination that help us to characterize what is happening in these individuals and then subsequently in the populations. Then we draw a blood sample, and we've been able to characterize lots of different normals caused by that, kidney, liver function, inflammation, vitamin levels, proteins in the blood, and random and very specific impacts of blood tests that tell us about overall health. And then we can relate it through modeling to what else is happening in those animals. And how does a turtle that's really active, how do we expect its blood work to look compared to an animal that is really quiet? We then collect a swab, and in reptiles in particular, swabs are some of our best tools to look at health, because they can mask and be so many diseases and be very subtle. So swabs tell us about what's going on in their pathogens. So just like we would get a swab for strep throat, these guys don't get strep throat, but they get many other things. But at first, we didn't even know if a swab of the mouth was going to tell us what is going on. So we looked at the where to sample them, when to sample them. And then we start looking at, are there multiple things going on at once? And we find things that we'll talk about later, where we have some pathogens that are more common in the spring and others that are more common in the fall, and within the same group of individuals. And then we also can see presence of pathogens of zoonotic concern, things like Coxiella, which causes Q fever in other species in mammals. Salmonella, which is really uncommon. Less than 2% of turtles in the wild have ever had salmonella, and in most places in Illinois, we find it in less than 0.2%. Or things that cause cancer. So we've seen some viruses that are leading to cancer in turtles. And so all of these things kind of lead to what this looks like, in that we collect a lot of data, and we do a lot of different standardized measurements and that tell us about the health. And in total, every single turtle I look at, I record about 180 different parameters. I spend a lot of time on turtles. We spend a lot of money on turtles, because again, their value to the ecosystem and to the environment is so high that we can gather a lot of different information. But that doesn't make it easy. Turtles, because they're long-lived, they don't reproduce until they're at least 12 to 18 years of age. So we don't even go through one generation usually in a career. So the turtles that I started with, that were babies when I started this 17 years ago, Some of those are not even reached adulthood and produced the next generation. And so this is a multi-generational project. I try to get lots of kids involved and excited about this because I'm going to have to pass it off to somebody, even though I will be doing this for the next 20 to 30 years of my life. It's not, it doesn't tell us everything really quickly. So what does this lead to? I have my PhD in epidemiology, and so I track the trends in health and pathogens and disease. And so we perform all of these health assessments where we're looking at physical exams, the demographics or morphometrics measurements, blood work, infectious disease testing, and we know they all impact each other. And what we're trying to do, instead of trying to find things that cause disease and avoid that, right? That's kind of reactionary medicine. Honestly, it's a lot of what we do and is done in human medicine is that you go to the doctor when something's ill, and then the doctor tries to figure out what it is, and it will either treat it, but it doesn't necessarily prevent it from happening. And again, and it doesn't tell you if and when it's going to happen again. Or if you visit the doctor 1 week, they may tell you everything looks good, but then 2 weeks later, let's just say you get sick or you get cancer, and it wasn't showing up 2 weeks in advance. So it's not very predictive to tell you when you're going to get sick. So we're trying to flip that. We're trying to take all of these interactions with turtles, and instead of trying to avoid disease, which is what most of medicine is, we're trying to promote health and promote wellness. So we developed this Wellness of Wildlife project, and we're just really flipping the paradigm. But that's obviously not something that is quite easy because you can support wellness and do things that help to prevent illnesses in humans like cardiovascular disease, but you can't promote overall entire wellness to prevent all ill effects. And that's kind of what we're trying to do from a population standpoint with turtles. And so how do we do this, and how did we start this before we talk a little bit about diseases? Um, is oftentimes in wildlife medicine, you are worried about one disease. You whether it's mycobacterium or Ebola or any of these things, you tend to just focus on one thing and be very tunnel visioned. And in turtles, it's things like Ranavirus or Mitomyces. This is a shell fungal disease. This is a virus like Ebola that we're going to talk about next. And so you just focus all on that one pathogen. But then you start to realize, okay, it's not just about one thing. There are multiple things going on. And so you start to figure out, well, what else is out there? What else is affecting these turtles in addition to just that one thing, which leads to looking at multiple pathogens at a time. And that's what we do with box turtles, and we'll talk about it. And then this whole process, every day, every year, we refine and retest in order to improve our ability to say if a turtle is healthy or not. It might be surprising, or maybe it won't be, that after 5,000 turtles, if somebody brought me a turtle and asked me if it was healthy or not, I still could not tell you whether it's healthy or not. That's how difficult and subtle these changes are for turtles, and how our tests really have not allowed us to be able to truly tell whether an animal is healthy or not. I can tell you if it's got a pathogen or a disease, if it's got kidney disease or inflammation, but again, I can't say, is this healthy or not? So let's talk about some diseases that affect turtles in Illinois and on the landscape. So we've got Ranavirus. Rana is the derivation is from a frog. There's genus Rana. And so this is a disease that affects lots of different species. It's a double-stranded DNA virus. It cannot survive above 31 degrees C. So that is right around in the mid to upper 80s. Um, and and so this is why mammals and birds are not affected by this disease at all. But box turtles are. 90% of box turtles that get this disease die. It is incredibly invasive. But unfortunately, all reptiles, amphibians, and fish are susceptible. There was an outbreak at Kickapoo here in central Illinois that we had multiple species with 7 different amphibians, in addition to the box turtles, that were affected over about an 8-week period of time where there was about 6,000 dead individuals. Some are more susceptible than others. There are some species that are really quite resistant to it. And it likely lives in the environment, in the soil or water, or within a reservoir carrier animal, like a leopard frog, northern leopard frog, or um, a, a bullfrog. Um, the hosts were talking to me about that your next kind of seminar series is looking at invasive species. And so it's probably not surprising to hear that northern leopard frogs and bullfrogs are invasive in many places um, in Illinois, and they're carrying diseases that are potentially going to kill native species. One of the reasons I love working with reptiles is like they do everything slowly. Like absolutely, it's wonderful. Oftentimes, as a veterinarian, I have my time with reptiles in order to figure out what's going on. And in fact, sometimes I can figure out what's going on and start treatment, and then a month later realize, okay, no, I looked at that wrong, and I go back and I fix it, and the turtle didn't even know I did anything wrong in the first place. Well, that doesn't happen with ranavirus in turtles. Rapid course of disease. It's the one thing that will cause a turtle to get sick and die quickly. And in fact, it happens so quickly that the median survival time from a completely healthy animal to when it's dead is 21 days. Like it is quick. So this is very similar to Ebola. But that's in adults. In juveniles, it's even faster. Less than 7 days from when a healthy turtle gets exposed to this virus to when it's dead, median survival time. And it's across all different species. And you know how I said before that it's really difficult to tell whether a turtle is healthy or not. So all of our usual techniques that are used for mammals, certain blood tests are not terribly reliable. Um, but one of the things that we were able to develop that I had a PhD student, he's a veterinarian, and he was doing both degrees. Um, we looked at cytokines, and what we found was that cytokines are overexpressed and overactivated in turtles that have ranavirus to a point where it leads to the cytokine storm. And that's very similar to probably things that you've heard about in COVID, where this same mechanism is happening in turtles, but it's temperature dependent. So turtles that are maintained at warmer temperatures go through the cytokine storm and die quicker and more robustly than ones that are at cooler temperatures. And then one of the interesting things about ranavirus is that, ranavirus doesn't live in a bubble. There are other things that affect ranavirus and exist in ranavirus. And what we saw was that we have 2 other diseases that are commonly seen in turtles, box turtles, that don't necessarily cause death, but they can cause some illness. And those are a herpes virus, which we're going to talk about, and a mycoplasma, which we're also going to talk about. But when turtles get exposed to ranavirus and they already have mycoplasma and herpes, they have a better survival than if they didn't have herpes or mycoplasma. So I'll say that again. Turtles that had more pathogens actually did better and survived compared to if they were just had ranavirus and nothing else. And I think that surprised a lot of my colleagues, and it surprises other people when I talk about that more 15 years ago than it does now, because we now know the microbiome and the virome within a person or within an animal is so impactful for wellness. And so it's not surprising that when you have a balance of organisms, that they're not necessarily as impactful as if you just have one, right? It's a monoculture versus a diverse culture of plants or animals, but in this case, it's pathogens. One of the frustrating parts about ranavirus, in addition to it being 90% mortality rate, is that and we've had opportunities where we've really experimented and tried lots of different things to save turtles with ranavirus. And so we've raised the temperature, we've given them antiviral medications, we've done lots of things, and we can save some turtles. What's unfortunate is that all of the turtles that survive become carriers and shed millions of copies back into the environment. So this comes into play with rehabilitation. I don't do wildlife rehabilitation. I work very much in the wild, looking at populations of free-ranging wildlife, but I work with colleagues that do rehab. But turtles that have ranavirus and recover can never be released back into the wild because now you've just created a carrier animal that can be in that environment for 100 or more years, and then kill many more animals. So if it isn't bad enough that it kills most animals that get it, even the ones that survive are never going to be part of a population ever again. So um, clearly, I'm leaving, a doom and gloom type scenario here, right? This is why I work so hard to make healthy animals, is because some of these diseases are really difficult. And as urbanization and suburbanization has occurred, right, we've had more roadways and that need to be put in. And so the natural environments really change, or even in the cases of really prospective thinking. So there was a case in Maryland and a case in Indiana where they were doing a highway expansion. And so they had biologists go in there and they collected all the turtles and they were putting them into a new environment a forested area that supports turtles, or they were moving them temporarily and then they were going to put them back with mitigation in those areas. Well, unfortunately, the two times they tried that, what they realized was you can't move turtles willy-nilly, and all of these animals died. In fact, this was 360 turtles that were moved. Within 3 months, 330 of those turtles were dead. So it is really aggressive. And then when you move them to a different location, it not only are those animals at risk, but if you move animals with ranavirus, then you're exposing every animal in that environment. So you can't just move animals and move them back or move them at all to good habitats, because you're transporting diseases. And this is hard. I've had family members, that have picked up wild animals and reptiles in particular, and be like, "Oh, I know this really good pond in my neighborhood. I'm going to release it there." Well, actually, you're probably transmitting and transferring disease that is more likely to kill animals than it is to save that one animal. So let's talk a little bit about some of the other ones, things like herpes viruses. That's a virus that family that you guys are probably familiar with, right? It causes cold sores in humans. And it's similar in turtles in that it's a double-stranded DNA virus, doesn't survive very well outside the body. But one of the things that's interesting, as we talked about ranavirus, it can kill everything that it really comes into contact with. Well, herpes viruses are unique for each species. So there are actually 3 box turtle herpes viruses, and box turtle herpes viruses don't affect people. And our herpes virus doesn't affect box turtles. And a Blanding's turtle, which is found in northern Illinois that we work with, have 2 herpes viruses, and those don't cause problems in box turtles. So even among turtles, there are differences. And there are differences between locations. Some have really negative impacts in that this was a herpes virus that led to cancer, and a squamous cell carcinoma. And this, again, was a free-ranging wild animal. Herpes viruses never go away, right? They're lifelong infections. But what we notice in populations is that there are times a year where herpes virus is shed by turtles. And so in May, you can see these black dots are where herpes virus positive animals are on this kind of ridge system. And in July, all the positives are here. And then in September, there are no positives. And then this is replicated the following year. And so there are environmental characteristics in these locations at this time of year where it promotes herpes virus prevalence and presence, but it doesn't necessarily kill these animals. And so it's not as big of a concern about releasing animals like it is for ranavirus, is because most places have that have turtles, box turtles will also have a box turtle herpes, and so you're usually releasing them. in similar planes of health. Mycoplasma or mycoplasmopsis is its new name, is a bacteria in turtles. Again, doesn't survive well outside of the body. And there is one mycoplasma among all the different freshwater turtles. So box turtles, Blanding's turtles, spotted turtles, all of these turtles have a very similar mycoplasma. And it can cause some significant effects, but most of the time it causes runny nose and lethargy. It is fatal in about 15 to 20% of the time, which is still a lot, but most animals recover. But it is seasonal. And so you have mycoplasma, which is this which is this dotted line, and it's low in the spring, really low in the summer, and in the fall, that's when we start to see all of our mycoplasma. And we see that again with lots of different things with reptiles. Similar to herpes virus, these animals, you should make sure that when you're releasing them, you're releasing a positive animal to a positive environment or a negative animal to a negative environment. You don't want to crisscross. And then the last virus that we'll really talk about quickly is an adenovirus. Adenoviruses are in people. They're in mammals. One of the vaccines that you get for your dog is in the distemper vaccine is an adenovirus. It's a double-stranded DNA virus. Lots of things get them. And most of the time, they're very specific for the animals that you're looking at. But there's a lot of diversity in adenovirus. In fact, we have more adenoviruses in turtles than in Illinois than we have of any other virus group in Illinois, or really in North America. It's also seasonal, very similar to mycoplasma. And this is Blanding's turtles. We have long-standing projects in Chicagoland region, and over the course of about a 5 sampling season year, we detected 6 different adenoviruses in Blanding's turtles alone. And some of them, as you can see, are rodent or passerine, bird, songbird adenoviruses. And so Blanding's turtles are omnivorous, and they will eat carrion. They'll scavenge. So likely these adenoviruses, which are really stable in the environment, were picked up because a Blanding's turtle was eating one of these animals that had died in their environment. Adenoviruses are really all around us. The more we do with adenoviruses, the more I realize that it is one of the most diverse groups that we have. And one of the things we haven't really talked about much is that just like they're in for humans and for dogs, there are diseases that you expect to happen in juveniles or kids, and ones that you expect to happen in adults. Well, that also happens with turtles in that Blandings for this one adenovirus, the majority of the infections are occurring in subadults. That is when they're most affected. But in painted turtles, it's adults. And in red-eared slider, it's adults, although red-eared sliders, they really just have a lot. Red-eared sliders are one of the species that are released non-natives in most parts of the northern state of Illinois. These are the most common in the pet trade, and so they often get dumped into rivers or streams, and they have lots of diseases when they do that, and they're bringing lots of diseases and threatening many of our native species. It's one of the things that I'm actively working on every year in Chicagoland is to look at those impacts and try to lessen those impacts. If you don't remember anything today, don't release a red-eared slider, and don't let anyone release a red-eared slider, especially in northern Illinois. These animals are really have lots of diseases and lots of pathogens that don't necessarily cause any disease or death in red-eared sliders, but in lots of other species they do. All right, so where does that leave us? It leaves us with like a big table that again didn't exist before we started this work, which lists all the viruses in the major species that we see in North America and where they are. All of the unknowns are gaps that hopefully over the course of my career we will fill in those gaps. And then we start to describe the epidemiology. but there's lots of things that are happening simultaneously, and pathogens that are occurring. So one of the things that we've been able to develop at Illinois and utilize in order to look at multiple pathogens at the same time, something called Fluidigm. It's a thermocycler that uses a chip, and we can test 88 samples plus the controls with in triplicate simultaneously on a small volume of sample. And it gives us this heat map. The lighter, the hotter it is, means that more positive a sample is. And so we can look for every different turtle. This is how we can do multi-pathogen work. We can look at about 2,900 different PCR reactions in a single run. And it gives us lots of data about what are these interactions. And so how does this look like? When I first started in 2017, again, just similar to what we first talked about, I only looked at Ranavirus, which is frog virus 3 is the acronym here. And then we started to add herpes and mycoplasma and adenovirus. And then when Fluidigm came online, look at all of these pathogens that we can look at simultaneously in order to characterize the health of turtles. It is it's really been an amazing tool for us in order to apply this across multiple species. We do this now in gopher tortoises and desert tortoises, blandings, spotted turtles, paints, snapping turtles, you name it. But then we can boil it down, especially for our land managers at the DNR, into pathogens categories, things of high threat. After we've done this, what are the ones that we really need to keep out of our wild populations? Ranavirus, Amydomyces in Illinois for sure. Things that are host adapted, meaning they only cause a problem if they are um, really get in a bad host or an abnormal host, but in general, they don't cause any problems. Things like herpes viruses and mycoplasmas, adenoviruses, things that we just don't know what their significance is, specifically Sulawesi tortoise adenovirus. This is a adenovirus that I learned about 20 years ago when I was doing my board exam that had killed 100 Sulawesi tortoises within 3 weeks when they arrived in Florida. And throughout the last 20 years, zoo veterinarians are like, we can't have Sulawesi tortoise adenovirus. Well, nobody had been looking for it until we started looking for it in Illinois and realized, well, it's in painted turtles, and those painted turtles are doing just fine. So what likely happened is that Sulawesi tortoises came to Florida because they're naturally from Sulawesi and not from here. And they got exposed to normal painted turtles from North America. And the painted turtles have Sulawesi tortoise adenovirus, and they gave them the Sulawesi tortoises new virus that they'd never seen before, and they all died. So again, moving turtles even if they look normal and healthy, normal, you don't know what they're carrying, and they're likely carrying something that may not cause a problem in them, but is causing a problem in something else. Things that are associated with cancer, mainly herpes viruses, and things that affect people, things like lepto and Coxiella, salmonella. So this is really helpful. We're going to end with kind of an important topic here, where we're kind of in a period for turtles where there's an epidemic occurring of confiscations and animals that are being pulled from the wild in North America and heading to Southeast Asia for various different reasons, pet trade, medicinal use, numerous different things, but thousands of turtles affected. And those turtles then are usually confiscated if they don't make it in these large airports, and they're stuffed in socks or shoes, or in some cases, they're wrapped in duct tape in order to get there with really poor husbandry and welfare considerations. But I come in when we talk about health. And so there was in 2021, there was a confiscation of 100 turtles that went to some of our partners in the zoo field. And within 3 weeks, 40 turtles died. We started doing testing, and it was the most complex pathogen profile we've ever seen. So you know how I said that, we had all of those turtles or all of those diseases, most of them are not very common, less than 10% of the time. And Ranavirus, it's really pretty rare. Well, over half the turtles had all of the viruses and bacteria that we test for all at the same time. But everybody had at least two. And so we took the remaining turtles and we tested them for all of our different fluidine pathogens. We brought them to the University of Illinois. All of my veterinary students and residents and PhD students, we all participated, and there were lots of different things. And these turtles don't have this in the wild, right? These turtles got this because of these people, these collectors that took them in and then concentrated them in unnatural conditions. And then there was another outbreak or another confiscation just of 17 turtles. Um, and we found that these pathogens are lifelong and they shed them abnormally for what a normal turtle would do in the wild, right? We saw how herpes viruses, really are shed in one location in May, one in July, and then not in September. Well, these turtles have completely disrupted their normal physiology, and they're shedding these viruses and bacteria year round. And in some cases, their prevalence rates are 90% of the turtles in any one month. And then, but none of these turtles, many of these turtles don't look sick, which is great, except now we can't put these turtles back in the wild because they've got all of these diseases in much higher numbers than they used to or would otherwise. So this leads me to kind of my roadmap for wellness and the wellness of wildlife is that long ago we got rid of the single pathogen surveys. They're really insufficient. We need to add multiple things at the same time and use multiple modalities. Like you can't just use one thing. You can't just look at blood. You can't just look at swabs. You can't just look at physical exam. You can't look at a turtle and know it's healthy or not. Um, and so you do this initial response, usually in response to a threat like Ranavirus. Then you add components, blood work, habitat comparisons, pathogens. You perform the epidemiology, and then you manage the wild populations so that they're better and that there's less impact of these pathogens. There's lots of different funding sources, and then really the bulk of my work, and I'm really fortunate to work at a vet school that has lots of eager, albeit type A individuals like myself that are really interested in saving the world and finding ways to do that through understanding what's going on with these turtles. And so we have lots of different people in lots of different scenarios to help. And these go on to doing amazing things. Veterinarians at many different zoos, private practice. Those of you in Chicagoland may recognize some of these individuals. And this is what is truly wonderful about this program is it involves so many different people. We start them young. This is my daughter. And there's an impact just beyond doing the science. It's also training that next generation of conservationists to make an impact and make a difference by doing things that are really done anywhere else in the world. So with that, I'm going to end my talk, and I am happy to take some questions. Thank you.
Wonderful. Thanks so much, Matt. That was fantastic. A ton of great information and really fascinating to hear. So thank you for all of that. I'm going to go ahead and start reading some of the questions in the chat. If you have a question, please put it into the chat and we will get to those. Um, so we had some questions on the Ranavirus. Can you talk a little bit about like how does a turtle contract Ranavirus? Like how does that disease transmission happen?
Yeah, I'll be honest, this is the scary part. So if we're going to keep the comparison with Ebola, right? Like it's, short duration of time, causes massive mortality, hemorrhaging for sure. But Ebola has to be transmitted directly through secretions. And so that usually outbreaks can be controlled with biosecurity. Ranavirus is transmitted most often in the water. So turtles are often the aberrant or dead end hosts, meaning that they're not the target. They go to water that's infected, and they get in they get the disease there. But oftentimes amphibians are the amplifying hosts. So we've got lots of frogs in ephemeral ponds or salamanders, as the case may be. Those animals die rapidly as well and release millions of copies into the water. And then anything that visits that water during that period of time will then become infected.
Okay. Speaking of disease transmission, if you are handling a turtle, can humans contract some of these diseases?
None of the ones I talked about today.
Okay.
So, there's some zoonotic ones that I didn't really spend any time with, right? Salmonella, lepto, Coxiella, which we find in Illinois turtles rarely, that people can get, but not any of the ones we talked about.
Okay. Are any of these viruses passed on to the embryos in the eggs?
Great question, and one that we don't quite know. So, Ranavirus probably happens so quickly that it wouldn't. It kills animals so quickly. I guess in a surviving animal, it's possible, um, but that is very difficult to track.
Mhm.
Um, Mycoplasma is interesting. So everything gets mycoplasma. People get mycoplasma, and it causes different things in different animals. Most of the time, it's respiratory, but there's a lot of times mycoplasma causes joint pain and swelling. Like in raccoons, that's like the biggest thing. Um, and in turkeys, mycoplasma causes like kind of swelling of the eyes. And what they found with, and it's a particular mycoplasma, it's not one that we get, but um, what they found with that mycoplasma is that it is passed in the egg. Um, and and so if mycoplasmas can be passed in the egg of birds, it's likely that that's still possible for turtles as well.
Okay. Um, so I asked, can humans contract a disease from the turtles,
Mhm.
but can turtles contract a disease from humans if humans are handling them? Can they get-
Yes. Those are usually topical,
Okay.
um, and usually associated with bacterial transfer, like into wounds.
Mhm.
Can they get flu or COVID from a human to a turtle? We have not seen evidence of that. We've tested turtles for COVID and we haven't found it in turtles. Um, Humans could give turtles salmonella um, if a human um, was positive,
Mhm.
but that pathway is not really well established. It
Okay.
is possible, but it's pretty rare,
Mhm.
uh, and we don't see it. We actually don't look for it, actually.
Okay. Um, this came back from the beginning of your presentation. It was just a question on, you talked about how there was such a low percentage of turtles that get salmonella, but that kind of association is there that we feel like a lot of them have. Do you know like where that perception came from or like kind of the background on that?
Yeah, this is one of the silliest things in public health that I've experienced. And I am a staunch advocate for public health. If it wasn't obvious before 5 years ago, we need better mechanisms in order to protect the public. Um, and 75% of diseases of humans come from animals, and two-thirds of that are coming from wildlife. So but this is odd. So there is a CDC recommendation that goes back to the 70s that turtles can pass salmonella. Well, turtles can certainly have salmonella, and it's pretty rare. But the reason that it got associated with that was, and this was back when, my mom was a kid, because it was even before me, where they would sell those small little sliders in pet stores.
Mhm.
In Illinois and in the most of the United States, there's a law against selling any turtle less than 4 inches in length. But the reason that salmonella is associated with turtles and reptiles is because 4 inches or smaller is associated with the size that of a turtle that can fit into the mouth of a kid. I that is the exact reason that 4 inches was used is because they measured kids' mouths and they're like, well, 4 inches, they can't fit it in their mouth to I don't know, suck on it, or I'm not sure what the kids do, but I've got two of them, but they don't do that. Um, and so, but it's not based on the science because, to be honest, chickens have a much higher salmonella than prevalence than any turtles do.
Fascinating. I have heard that before now that you say it. This is ringing a bell.
Yep.
Fascinating. So looking at the red-eared sliders for a second, we had a couple questions. Do you know if there like why aren't there any regulations like limiting selling them from pet shops so they're not being dumped? And or is there movement towards that? Can you speak on that a little bit?
Yeah. So this one's variable. So all laws for wildlife are state by state. So there are certain states and several states, especially in the east and surprisingly some in the southeast, that prevent the selling of red-eared sliders. Illinois is not one of them. One of the problems is that um, there is it comes into ownership as well of turtles. There are some states that completely prevent the ownership of native species in that state. So, like in Tennessee, you can't own a box turtle, even if you got it from a captive breeding facility, because box turtles live in Tennessee. Illinois does not have that law. And in southern Illinois, red-eared sliders are in their native range.
Mhm.
But in northern Illinois, they're not. And so that's why we can't regulate based on the location within the state because it would be, I mean, it would be impossible to enforce. So it really is unfortunate because I'm doing the back end and seeing the impacts of this. The DNR is doing the back end of this and trying to regulate the release, but really the only thing we can do is take once we find them, we take them out of the wild. And oftentimes we euthanize sliders if they're not in their normal habitat.
Okay. Um, so this one question had to do if there is, say, a park district where people have continuously been abandoning their sliders into that pond, there's a huge colony, it keeps expanding. Can anything be done, or is it just kind of just keep them there and try to discourage further, dumping of
So, uh,
animals? What do we do about that?
yeah. Um, I mean, this is multi-faceted answer,
Mhm,
right? Like it's not just one single answer. Um, the because you've got to try to prevent the behavior while also dealing with the current scenario. And both of those are a little bit different. If there is a way to prevent release through education or exclusion,
mhm,
well, then that opens up the door in order to then figure out what we're going to do with that population and whether or not that is containing that population so it can't move back and forth and they can't contribute to viral or bacterial or pathogen spread. That's certainly an option, although it's not quite an option. Or it is a completely deconstructing the habitat and all of the animals in them, and then kind of starting over. It is unfortunate. Like as a veterinarian, I certainly have the option for euthanasia, and euthanasia is the hardest thing I do. Even in species that aren't supposed to be where they're at, it should be hard. If it's not hard for me, then I don't want to be a veterinarian anymore.
mhm.
But in this scenario, it's not the worst thing I have to do. Pre Euthanizing invasive species is a really unfortunate thing that I have to do because of our society and humans that have either advertently or inadvertently contributed to it. But it's preventing the release and the destruction of native animals and likely unseen deaths that are occurring because of those. So while I don't like euthanasia, to do euthanasia because it's hard, I very much like to do it when it's incredibly important to protect animals.
Yeah. It's a great perspective. Thanks for sharing that. Um, there's so many great questions coming in. I'm gonna try to pronounce this one. Have you done any work on Emydomyces.
Mitomyces, yeah. So yeah, well, I spend a lot of my time on Mitomyces right now. Mitomyces is a called turtle shell fungal disease, and it is mainly in the western pond turtles in the Northwest. It is not common in Illinois, although we test 12 to 1400 animals every year for it, but we are actively trying to prevent its release. We found it in a population of Blanding's turtles that were being head started, and it's completely derailed some of our conservation plans in the state. But we found it because we were looking, and now we've prevented the release of positive animals into the wild. We've been working on lots of different treatments. We did 10 treatment trials involving 300 turtles. I've got lots of stuff that we're doing with Mitomyces, but we are we are actively this is the example of how our work is directly impacting conservation on the ground because we are preventing the release and saving animals from being released infected.
That's great. Well, there's more questions in the chat, but I know you have a class to teach it too, so I'm gonna wrap us up. For those who still have their questions not answered, I'm sure Matt wouldn't mind if you sent him an email, could get back to you in that way. Thank you so much, Matt. Great talk. Lots of wonderful information. Thank you everyone for interacting and for those great questions. We really appreciate you all. And like I said, we will get this recording up on YouTube and share it out with you. Thanks, everyone, and we'll see you for our next webinar in a couple of months. Take care.
All right, thanks everybody.